From Microbe TV, this is TWiP, This Week in Parasitism, episode 260, recorded on June 6, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about parasites. Joining me today from New York, Daniel Griffin.
Hello, everyone. I'm actually, tonight, I don't know if I mentioned this, Christine, I'm getting on a night flight to Edinburgh.
Ooh.
And then I'm going to get on a train to Inverness. Then I'm going to rent a car after spending the night in Inverness with my wife and Daisy and Barnaby. And I'm going to head to the Inner Hebrides, and then I'm going to drive and then take another boat to the Outer Hebrides. Going to go all the way to the northwest corner of Lewis Island. And then apparently, if that's not remote enough, another 2.5 hours out to some remote uninhabited island that my wife has found. So,
why do you want to do this?
I love this kind of stuff.
Sounds amazing.
Okay. Well, you're supposed to do a podcast next Thursday. Are you going to be remote?
We're going to have to see how that works. I don't know. Maybe there's some way I can, we'll see what happens.
Also joining us from Glasgow, Scotland, Christina Nowla.
Hello, everybody.
What was I going to say before Daniel went on that long thing? Oh, first of all, yesterday, June 5th, was the birth anniversary of Dixon des Pommiers. So we want to note that long-standing founding member of TWiP and TWiV.
And Parasites Without Borders.
Parasites Without Borders. So he would have been 85 years old yesterday. We miss Dixon a lot.
We do. And that's why I'm wearing my yellow jumper, because he used to call that my bee jumper.
That's funny.
And what also is today, right? Today's another memorable day.
Today is the 81st anniversary of D-Day. Right.
Wow.
And the Allies mounted this huge invasion on the Normandy coast. Big thing. And we were just saying that I'm not sure that the U.S. would do that. Now the U.S. has divorced itself from Europe, apparently.
I thought it was a really good comment by, it was the representative from Germany that was in the Oval Office recently, and that came up, and the suggestion was, that was a tough day for you guys. He's actually, no, tremendous day. That was the day that we, the Germans, were liberated from fascism.
Mr. President, this was the liberation of my country from Nazi dictatorship.
Yes.
Interesting perspective on that.
Yeah, that was the beginning, right? The troops made their way to Berlin.
Yeah, the start of the liberation. Yeah, there was still a very long road, and a lot of people sacrificed to get us.
Just a lot of people died on D-Day. I don't know if you've been there, but there's a wonderful cemetery up there right near the beaches where they landed, and it's just rows and rows of crosses, right? Something like 12,000 people are buried there.
Wow.
And France donated the land to the U.S. In memoriam, So yes, that's D-Day, June 6th.
And it's TWiP Day. That's also a good thing.
Today is TWiP Day. And
yes,
if you enjoy these programs, Microbe TV, all the podcasts, we would love to have your support. We don't do advertising. We depend on listeners to support us. With as little or as much as you can, go to microbe.tv/contribute. We are telling you real science, no lies, no misinformation. So we need your help to do that. Microbe.tv/contribute. Now, we made a mistake on our last TWiP. We forgot to give away a book.
At least it was only that.
Yes. First time ever. And I wonder if people who listen to the clinical update also listen to the regular TWiPs, the non-clinical updates.
There's some. I think the clinical update audience is larger.
Bigger, yeah, probably.
Which is switched, right? I remember the good old days when TWiP was like the most listened to.
Well, we didn't have a clinical update till you showed up.
Yeah.
Right?
Yep. Then it got really popular for a while, and then the pandemic really launched TWiV popularity. So.
Oh yeah, big time. Yeah. Should I hope for another pandemic? No.
No, but I guess we can rectify the book issue.
Yes, let's pick a book. We had lots of listeners last time, and let's see how many. What's the top number? 11. 12, 13, 14, 15, 16. Wow, a lot. 17, 18, 19. Oh my gosh, 20 eligible listeners. I'm going to go to random number generator.
And that's probably why we forgot, because there were so many, we were exhausted.
I've already forgot how many. 20.
22, I think he said.
Did I?
I think so. Ooh.
20. So we're going to pick a number between 1 and 20. Are you ready? Dixon used to do a drum roll. 11.
All right.
Who is our winner? Let's scroll back up. And it is Joseph from Cut and Shoot, Texas. That's cool. Joseph.
I remember. Yep.
You are our winner. I hope you listen. He's been listening since 2016. Okay, good. Send your address to TWiP at microbe.tv. That's an email address. And just put in the subject line, TWiP 259 book winner. And since you're in the U.S., we don't need your phone number. And we will ship you out a book post-haste. We have quite a few left with yours and Dixon's autograph, Daniel.
That is true. Yeah. When I was last in, we went through, we realized there was still a number of those precious books.
Joseph, congratulations, and send us your information. All right. Today is TWiP Day with a paper. And this is being done by Christina. Tell us what you have, Christina.
Yeah, so I chose a paper in the context of malaria transmission, and in particular, kind of at the crossing between the mosquito and the human, or another mammal, I suppose. So this is a paper from Nature Microbiology, and the title is Parasite and Vector Circadian Clocks Mediate Efficient Malaria Transmission. So we have 3 first authors. These are Ines Bento, Brianna Parrington, and Rushlin Pascual. And I think the corresponding author is Rio Ferreira. I actually didn't write down the first name, so I apologize for that. And they come.
Philippa.
Okay, thank you, Philippa. So they come from lots and lots of places. So we've got the Gulbenkian Institute for Molecular Medicine in Lisbon, Berkeley Public Health, University of California. We've got Jan Zuckerberg Biohub in San Francisco, Department of Neuroscience in the Southwestern Medical Center in Dallas, Texas, the Howard Hughes Medical Institute at the University of Texas Southwestern, and then the Faculty of Medicine of the University of Lisbon in Portugal. So, a fair spread of authors. So, yeah, so this paper is going to be looking at the transmission from the mosquito into the mammal host. And I thought maybe before I get started, because we don't often kind of look at the life cycle part in the mosquito, and I know it's maybe not extremely relevant, but I thought it would be a good opportunity to maybe mention that actually the parasite does undergo quite a lot of development within the mosquito host. So I'll just gonna maybe take you through that part of the life cycle. So when the mosquito takes a blood meal, there will be macrogametocytes, and microgametocytes will be taken up by the mosquito, and the microgamete kind of comes out or exflagellates out of that banana-shaped entity, previously a microgamete, and fuses with the macrogamete, so that's the sexual part of this life cycle, to form a zygote. And then there's some replication going on, and eventually we end up with a eukainate. And this then moves across the midgut epithelium within the mosquito, and it actually looks a bit, when you look at pictures of it, it looks a bit like Cryptosporidium, and I don't know if you and our listeners are familiar with that. So the Cryptosporidium are intracellular, but not quite, so they're kind of sitting on top of that epithelium, and they're covered with, I suppose, with their plasma membrane, so they kind of develop into oocysts, and then sporogony happens, and eventually the sporocysts, they are called sporozoites. Sorry, I got confused there. The sporozoites then rupture out of those cysts, and they migrate to the salivary gland, and then they just hang about until a mosquito takes its next meal, and the sporozoites then get injected, and then they move on to infect liver cells. And it's really that kind of last step where that paper is sitting at in the life cycle. So
I was going to say it's, we I guess found this important enough that I think it's one of the only times when we include the insect part of the parasitic life cycle. And so, if people have a copy of Parasitic Diseases, the 7th edition, the 8th is hopefully going to be coming out pretty soon. I've given all the authors a 9-day from now deadline. So when I come back from the Outer Hebrides, they're supposed to have read everything and told me if we're ready to go. But, we don't even mention or spend much time on the fact in our life cycle, when the sporozoites go to the salivary glands, as we're going to learn in our paper, or people are maybe introduced to concept, they're there for a while. They're there for days. And what are they doing? Like, why go there for days? So it's pretty interesting.
Yeah, parasites are quite amazing. Trypanosomes also have a really quite complex life or development cycle within the mosquito, silly me, the tsetse fly. So maybe one day we could dedicate some time on exploring that in more detail.
We
for various
should.
parasites.
I love the tsetse fly.
Yeah, me too. They're such cool creatures.
There are some parasites that are not so cool.
Yes, yeah. I mean, on every parasite there is that horrible side. Like, why do we care about them? Because of the horrors they cause. But they are fascinating and complex.
Yeah, and really, they have really clever life cycles and adapt to their environment really well. Anyway, so malaria transmission is dependent on the mosquito's behavior of seeking a blood meal. And that actually with Anopheles mosquitoes happens mostly in the night. So these are night feeders, and that's why insecticide-treated bed nets are so efficient at controlling malaria. Wouldn't be the same, for example, if we were talking about an arboviral infection like dengue or Zika, because the Aedes mosquitoes are kind of biting in the late afternoon before you snuggle up in bed. So a bed net is not very efficient there. So this is a kind of circadian rhythm-driven behavior that allows infected mosquitoes to deposit Plasmodium parasites into the skin at a specific time of day. And during a blood meal, these uninfected mosquitoes, they can also ingest transmissible parasite forms, of course, pick a new load up, so potentially from an infected host. And then what happens is what I just explained earlier. So, and at the beginning of the blood meal, the mosquito injects actually quite a lot of different components into the skin. For example, so these are saliva proteins, and they can impact parasite transmission by increasing the parasite load during the infection. But that's not something we're going to look at today. But I thought that was actually quite interesting as well.
Yeah, and Dixon used to refer to it as a salivary pharmacopoeia.
Right. Yeah. So, and then saliva ingested together with blood also modulates the parasite development in the midgut of the mosquito. So I guess that's when we go in the reverse way, not from the mosquito into the mammal host, but from the mammal into the mosquitoes. So, and it's thought that parasites can actually modulate the vector physiology during the infection, and some saliva proteins are really highly expressed in infected mosquitoes when we compare them to uninfected ones. And I think that that's really, that's not a lot of background, but I think that is really highlighting that it is quite important that we understand that kind of biology of the salivary glands, how that can maybe promote development of strategies for eventually combating this parasite. Although I suppose that's something that we always aim for, but are often quite far away from. But I suppose it reads really nicely on a grant proposal, doesn't it?
Right.
But I think there may be something here. I mean, this is really interesting. Like they observed over time this wonderful pharmacopoeia, but why? Is it just that the parasite takes advantage of what's there? Or, as the authors are going to suggest, and I think support, maybe the parasite has the ability to modulate that pharmacopoeia?
Yeah. So now let me get back to the kind of circadian rhythms that I mentioned earlier, for the kind of host-seeking behavior. So actually nearly all mammals display robust daily rhythms of physiology and behavior, and that's really quite well researched. And these are approximately 24-hour cycles. They're called circadian rhythms, and they are driven by a master clock, which in humans is found in the hypothalamus. And actually a lot of processes have these circadian rhythms, including some metabolic processes and also some immune function. I think the most obvious cycle driven by this clock is really the sleep-wake cycle, which is obviously critical for our health. So our listeners, you may be familiar with that. And actually, it's been shown previously that Anopheles mosquitoes, they also have transcripts with circadian rhythmic expressions, and they are probably controlled by core clock genes as they are in mammals, but we don't really know very much about that, not nearly as much as we do in mammals. But it is thought that these clock genes, along with kind of environmental cues, coordinate Anopheles pheromone synthesis and swarming and also mating behaviors. And maybe these clock genes, if they do indeed exist, like they do in mammals, they kind of maybe maintain the robustness and the precision of the mosquito's internal clock, and maybe ultimately influencing its feeding time and potentially the transmission of malaria parasites. This is actually the main content of that paper. So
let me ask you something. So, these mosquitoes prefer to bite at night, right? So malaria is quite successful. That implies that a lot of people are up at night and they get bitten, right? Or else they're in their homes and they don't have screens on their homes, right?
Yeah, I suppose also, malaria has been around for a long, long time, and, kind of these control tools that we have are really only recent when we kind of look at the evolutionary history of malaria.
Yeah.
So I suppose, were it to evolve today, it might maybe, I don't know, choose to be a day biter if we want to anthropomorphize evolution in that way.
It is interesting. Like, why bite at night? I mean, was the advantage that if you bite at night, you're maybe not going to get swatted and crushed? Is it, something about you need more time? Is there, something about the human at night? There's also the issue that, right, when someone's really sick with Plasmodium, they end up just basically laying there, so that the mosquitoes can feast on them. And I think we've discussed that before. But it is interesting because some of the mosquitoes that are involved in other transmission, viral, all the arboviral transmitters, the Aedes, some of those guys, they're crepuscular, right? Morning and evening. So I guess different. And then we've got Stephen's eye, which is now a daytime biter spreading malaria, right? Moving into urban centers.
Yep. Really interesting as well. Where were we? So we're talking about circadian rhythms, and actually mosquitoes also have circadian rhythm. But so this paper focused on salivary glands. So it was prior to that study not known whether mosquito salivary glands are also under circadian control, and whether maybe these rhythms influence the daily biology of the sporozoites inside the mosquito salivary glands. And, I think it's a reasonable hypothesis to have that this might be the case. And this is really, if you had these circadian rhythm or these kind of protein expression profile, that would maybe allow the anticipation and preparation for blood meals, so that the sporozoites would then be exposed to some signals within the mosquito to be ready to move on and hit the ground running really as soon as they enter the mammalian host. So in this study, I'm going to just give you the take-home message at the beginning, and I hope that's not really going to spoil it too much. But the authors describe the dynamic nature of the mosquito salivary gland transcriptome, so all the genes that are transcribed, and the relationship with the circadian clock. And they showed that actually a substantial proportion of genes that is involved in blood feeding in mosquitoes, including, for example, anti-blood clotting proteins, they do have a circadian expression within those salivary glands. And also, they also found that salivary gland sporozoites also have daily rhythmic transcriptomes, and particularly in genes that are associated with parasite transmission. So that's really probably sums up the paper, but I think it'll be worth our while going through the data in a little bit more detail. I hope you agree with me and don't just say, all right, let's call it a day.
It's gonna be interesting, right?
Yeah.
Because I led with the comment that, Anopheles gambii and some of the other Anopheles that are really responsible for, we'll say, P. falciparum in humans, they're nighttime biters, right? But they're going to use in this experiment, they're going to use Anopheles stevensii. They're going to use Plasmodium berghii. Yeah, so they're going to be using a different Plasmodium species. But I think my real sort of, comment right off is they're using stevensii. And I kind of want to know about this in gambii. Right, because that's the one that the big one for us. So this is interesting. The daytime biter has this, and then it's going to be interesting. Like as we're looking at transcriptome, that's great and all, but it's going to be interesting to know about timing relative to the actual protein production, which maybe we'll have the time to touch on as well. But
is stevensii a true day biter? I'm not actually familiar with that species in detail, because maybe that's just a recent thing as well, and that would explain why the experiments worked so beautifully. Yeah. Are you Googling that just now?
I'll check that as you start to go through. Yeah, as you start to go through the figures. And the data.
So and actually, maybe the findings could maybe be also it could be really useful in the context of other vector-borne infections, because many of those vectors have preferential feeding times. And we did, if you remember, we recently discussed the case of Brugia malayi infection, which is also mosquito transmitted, primarily Mansonia, but also some Anopheles. And you have to test for those microfilaria in the night, and that's where they're most present in the peripheral blood, and that's probably also to enable transmission or efficient transmission. So this might actually have quite far-reaching ramifications. Sorry, my words don't come. I've spoke French over the weekend, so still sometimes a French word pops into my mind. I was in Paris last weekend, so and when I was in Paris, the French words didn't pop in my head, but now they do. So it's really annoying. Anyway, so let's have a look at some of the figures in this paper and the results. And this is actually an open access paper, so I forgot to mention that. So if you're listening and you want to have a look, go and have a look at the link and the show notes, click on the paper, and you should be able to follow through, because sometimes it's quite nice to actually see the figures if you can. So in the first set of experiments, they showed that mosquito rhythms impact feeding and salivary gland biology. And so, pretty much all the experiments that they did were transcriptomic analyses. So they did the RNA sequencing, and they looked at the transcripts that were produced, and they did quantify and calculate and do complicated statistics. So this is really kind of the meat of the molecular experiments that they did. So in the first instance, they wanted to find out whether the salivary gland is under circadian control, and if that influences the feeding, essentially, and also the biology of the Plasmodium sporozoites. So they used as Daniel already mentioned, they used Anopheles stevensii, and they kept them under a strict 12 hour on, 12 hour off, light and dark cycle, or in a constant dark cycle. So they had 2 sets of mosquitoes for all these experiments, and I did that for 3 days.
Yeah, I was just Googling on the side and seeing mixed results, certainly urban feeders, but there was a study I just caught from Calcutta where saying about half of the female Anopheles were 54% was the number daytime biting. So interesting. I mean, but still we're seeing some pretty compelling data. So I'll put that on the shelf for the moment.
No, I think it's important to mention because that is maybe a recent shift. Who knows, because.
Yeah. And that would be interesting. Is that like an adaptive, and how long does it take for the mosquito to adapt to, let's say, a better opportunity, evolutionary pressure? Like, because it would be helpful to not just be restricted to nighttime and biting in the dark. And also the issue, like, we're using all these mitigation strategies where we have nets and they're impregnated, and we even put insecticides on the walls. And a lot of times the Anopheles like to bounce around. They like to land on the wall, and then they like to go here. And when they land on the wall, that can, get them exposed to the insecticide. So again, we're applying all this pressure. So interesting to know in these lab ones, or these ones that really are, given whatever, nighttime darkness feeders, or are they adjusting?
Yeah, I suppose, maybe these just didn't have time to adjust to daytime biting.
Yeah, and not every Anopheles stevensii is the same, right? Like one's in India, one's in Africa. There may be different. Yeah.
In fact, there is no light-dark cycle. It's all time-based here. That's the point.
Well, that's
right?
So they did do light-dark cycle, but actually it turned out it really didn't matter. Yeah. But we'll get to it. So actually, I forgot a really critical point. So these mosquitoes were all infected. I think that is quite important. That I should probably add that on. So after they subjected them to these dark light and dark cycles, then they dissected them every 4 hours over 3 days. So they did a lot of mosquito dissection. I've never done that myself. I've not worked with malaria, but I've seen people doing it. And it's a real skill, particularly if you have to pull out the salivary glands, I think. So what they found in that first experiment is that between 27 and 49% of salivary gland genes display a cyclic expression profile, which is a lot more than previously had been reported. Bearing in mind, though, that the previous experiments had actually looked at the entire head or the entire body. So there may have been, large parts of the mosquito is maybe not circadian controlled. So that may have kind of blunted that response a little bit. So that's a lot of genes. I mean, that's up to half of them. So when we now look at figures, this actually shows, figure 1b actually shows some examples of such transcripts.
No, it's really nice. Yeah. I mean, they've got, so, why do we skip past A? That's just sort of showing us.
Yeah, that's just the experimental setup.
What we're going to do. And then they're doing gene expression, right? Because, as we always say, gene expression, mRNA is not protein expression. We'll maybe get to that later. But you're seeing this really nice, 12 hour up, 12 hour down, 12 hour up, 12 hour down, right? So it's a wave, but basically every 24 hours, you're back at the peak. You go down 12 hours later, peak 24. So it's 0, you're at the peak, then you're bottom at 12, peak at 24, down at 36, up at 48. I mean, really nice cyclic expression.
And I suppose that's why they did the light, dark, and dark only, because they wanted to establish whether that was actually related to light. So what we can see here on the left-hand side is the mosquitoes that were only kept in the dark, and we can see the same cycling, really. And then in the mosquitoes that are kept at light and dark, we again see the same cycling. So that's actually a key feature of circadian rhythms, that they are maintained in the absence of environmental clues such as light. And what we really see here suggests that the genes are under circadian control, and that it is probably the time of day rather than the light that is the driver of those fluctuations in the mosquito salivary gland.
I think that's a good point, because the
Yeah.
first time I read this, I was like, oh, it's too bad that they didn't see this nice light dark. But, these are 4 or 5 day old mosquitoes. They have their blood meal. And then you want to look like, does it matter? Because otherwise you could say, oh, they're turned on by the light and off by the dark. Is the light stimuli doing this? Or, I think you point out very well, this is an intrinsic circadian thing that happens. It doesn't matter environmental.
Mm. So, we have transcriptional daily changes, which potentially might facilitate a successful blood meal. But for that, we really need to know what proteins are kind of cycling. I'll get to that in a moment. But first, I want to say actually that the genes that they're looking at that are transcribed in a cyclical manner here are actually well-known circadian clock genes. So 3 of those, I think, what were they called? Clock cycle,
period,
and Vrille. That doesn't sound very timely, but maybe that's some kind of time expression in a language that I'm not familiar with. Who knows? Gene names can be quite weird. And then I decided to look at figure 1F and 1G, and they really show the distribution of peak expression for all cyclic gene in the dark and the light dark mosquito. And actually, it looks like they actually have 2 peaks. That's maybe not so visible in the waveform figure that we see there, but if we look at them all, it looks all that most gene expression peaks at 4 hours after the light turns on, or at the beginning of daytime, and 4 hours after light's off. So it's kind of maybe 16 hours after the beginning of team time. And essentially, the salivary glands reach peak expression early in the day and night. I think they call this a double rush hour of gene expression, which I thought was quite funny. And you can see that in the figure F and G. And then really, they did quite a lot of other experiments as well, really to show that, some of these cycling genes actually, they had high amplitudes of difference. So some of them were maybe 2 to 3 times higher expression, but others were much, much more. There's some considerable differences in how much transcript is produced. So I think that's quite interesting as well. And then they looked at what kind of proteins were cycling, and that takes us on to figure 2 already. And that shows that most of the mosquito transcripts that encode for blood meal specific proteins, they cycle during the day, including, for example, genes that are important to stop coagulation of blood. So it's good to see that correlation. So it's not just any random old gene that is hanging about in the genome, but actually genes that are relevant for blood meals. And actually, it has been shown that in different experiments that when the expression of these genes is downregulated or knocked down, the efficiency of the mosquito blood meal has been shown to decline. So I think that's quite a nice control in there. So they single actually 3 genes out. They're called SAC, TRIO, and GILT. And these are associated with infection by malaria parasites. And just to give you one example of one of those, Saglin, which is encoded by SAC, who comes up with those names, binds the parasite microneme trap protein and is relevant for mid-gut parasite invasion and salivary gland presence. So, and the remaining figure 2 really show that not only genes that are important for blood feeding are cycling, but actually also genes that are involved in glycolysis and gluconeogenesis, so kind of metabolic pathways. And I do discuss that a little bit later, but I think it makes intuitively sense because when those mosquitoes eat, they also need to metabolize what they ingest. So I think that makes sense, and it's quite nice to see that as well. But overall, really, the data suggests that mosquito salivary glands have cyclical gene expression, and protein abundance rhythms that correlate with the feeding behavior in preparation for a blood meal, or maybe sugar metabolism, or blood metabolism. So that's the kind of first set that they looked at, and that's really kind of concluding the experiments where they looked at the salivary gland. And now we're going to move on to the sporozoites, the parasites. So in the next set of experiments, they show that salivary gland sporozoites actually also have rhythmic gene expression. And maybe before I move on, I just wanted to check if either of you wants to add anything or ask anything, or...
Yeah, I think, So far we've seen, and just gene expression, we don't know about protein yet. We've seen this cyclic gene expression. So the transcripts are going up and coming down, going up, coming down. But I guess now comes the burning question. Are the sporozoites involved? Is there any kind of correlation to the sporozoites? Are they, I guess, transcriptionally active? And are they showing any kind of a circadian rhythm that might be connected with what we've seen so far?
Yeah. I'm glad you mentioned the protein, because that just triggered my memory that I need to mention that as well. So we now have a good idea what happens with the mosquito salivary gland gene expression, not proteins. But so we'll look at the transcriptome of the sporozoites. And so this has been studied previously, and actually this stage is referred to as quiescent. So these sporozoites are thought to be cell cycle arrested. And it had been thought that they are transcriptionally similar across many days while they're sitting in the salivary glands. And I think that's something that is actually an understanding that's changing with that study. So, and actually the Plasmodium they looked at is Plasmodium berghei, which Daniel mentioned earlier. This is a rodent malaria parasite. And I suppose that is, it's just easier experimentally, I would think. So they actually probed the transcriptome at a very high temporal resolution, and they showed that about 12 to 20% of transcripts have a cyclic expression. And in addition, the sporozoites genes that cycle during the day have a maximum expression early in the morning with an approximate median threefold change in expression. So, and again, it's the same in both light conditions, so dark only and dark light. So that would again suggest that this is a true circadian rhythm, I suppose.
It's interesting because I'm still getting I know the answer because I read the whole paper, but at this point I'm like, well, does this make any sense? All these transcripts are going up early in the morning. Aren't we talking about night feeding? Like how is this even relevant, right? Is it the gene expression is going down at nighttime? I mean, we'll get to there, but just to sort of maybe people if they're falling asleep. They're listening to this, putting themselves at risk of getting bitten by Anopheles. Just to sort of put that, like, how are we going to explain that? What's going on?
I suppose they get transcripts ready to be transcribed when they're needed.
I think we're going to get the answer. We're going to get the answer.
So obviously the sporozoites are then injected into the skin of a host, and it's important that they can move so that they can cross the dermis and into the bloodstream and to reach and invade the liver cells, which is the first stop in a mammalian malaria life cycle. So they kind of use a substrate-dependent gliding motility that helps them do that. And there are many, many proteins that are important for that process. Mm-hmm. And if we move on to figure 3, in particular figure 3b, what they actually show, examples of sporozoite proteins that cycle, and some of these are involved in the motility of the sporozoites. So yeah, I think that is really quite nice. So, it makes sense to have more transcripts of that, so they can then translate it into proteins and move about. So actually they did some controls, because we thought that the sporozoites are thought to be cell cycle arrested. And I did some controls really to exclude that that cycling isn't really just a fluke of the sporozoites replicating in the salivary glands or more coming in at that one time. So they did a couple of experiments to check that. And I'm not going to go into any detail about it, but I think it's really good that they did that control so that we can be sure that those differences in parasite load did not account for gene expression changes that we see in this experiment. So this is actually a true transcript, a change in gene expression rather than there just being more sporozoites around.
It's interesting that they have a circadian transcriptome. I would think if I were designing a sporozoite, I'd have it react once it gets into the salivary gland, not to have it automatically going,
Yeah, I suppose that would be more efficient, but
maybe, I don't know, maybe it's too complicated to do the sensing.
Yeah, maybe. Yeah,
and just turn it on. But I would do it that way. I mean, I think it makes sense that the salivary gland does it. But
no, you're right.
Just thinking about the life cycle, right? So it ruptures out of the oocyst and goes right to the salivary gland. So that's really where it is.
Yeah.
So yeah,
the salivary gland has to do this because the mosquito feeds at a certain time. That makes perfect sense. But I would think that once the sporozoite gets into the environment of the salivary gland, there could be sensors that turn on whatever genes are needed for migration, as we see here, whatever. Yeah. But that's not the case. They're just turned on and off in a cyclic fashion. But it works, obviously. And my design of a sporozoite is not relevant.
Yeah, but maybe evolution is not always the most efficient. Or
no, for sure. Yeah.
So if we had a say, we would make it as efficient as possible.
Well, the problem is we look at things through a human lens.
Yes, absolutely.
And that's not always right.
Yeah. And actually, I just decided because these pictures in Figure 3, so G, H, and I, they're actually quite nice. And I said a moment ago I wasn't going to tell you what they did, but I think actually it's really quite nice how they looked for evidence of cell division of these salivary gland sporozoites. They couldn't find any, and they actually created a transgenic cell line that would allow the incorporation of a synthetic nucleoside analog of thymidine, and that would then bioluminesce. So they just kind of looked if this was built into the DNA in the salivary gland, you would obviously see the salivary glands light up. And I think that is really clever. And they didn't observe that at all. So they could be really confident that the sporozoites are not dividing and that therefore the cycling of transcripts is definitely not due to replicating sporozoites. So I think the take-home message for this section of the paper is that the transcriptional daily rhythms identified in sporozoites are not a consequence of cell division, but instead resemble a robust circadian rhythm. Also, they show that also being in a non-dividing stage, so they don't divide, sporocytes are actually transcriptionally active parasites with daily rhythms of gene expression that potentially allow the parasites to anticipate timing of a mosquito blood meal and prepare for efficient transmission, although not as efficient as Vincent's sporozoites, of course.
I don't know. I mean, it seems to be working. It seems to be working.
It works. No, it's a good, successful parasite.
Yeah. So now they actually kind of bring things together. So they're kind of looking at the daily rhythms of sporozoite transmission. So we're going to have a look at the rhythms of those. And so let me just skip to, I need to speed read my notes. So they did a functional analysis of the transcripts that were cycling. And they found that sporozoites have rhythmic, actually I already said that, didn't I? Transcription of motility-associated chains that were important for invasion. And I think I just wanted to give you some examples maybe. So they did actually show that more than 50% of microneme-associated genes of the parasite cycle throughout a day. And this matters because the micronemes, these are organelles that are located at the tip of the parasite, so at the apical end. That's why they're called apicomplexans, I think. I'm not sure, actually. Oh, I might just got myself into hot water. Moving on swiftly.
Moving on. No, you're fine.
So these proteins are important for invasion and motility. So the micronemes are full of those proteins and they release them. So they're secretory organelles. And they are fully packed with stuff that helps the parasites move and invade and infect new hosts. And one example is actually the circumsporozoite protein, which our listeners should be very familiar with. So essential for initial liver invasion. And obviously also not from the point of view of the mosquito or the parasite, but from a human perspective, that's also the target of the first successful malaria vaccine. And this is all shown in figure 4B, if you are still follow me with the figures. So you can see the cycling there. And actually figure 8 shows a summary of sorts of the functional analysis. So they grouping the transcripts by function. So I think that's interesting, but maybe less relevant overall. So that's all at the RNA level, and I'm glad why Daniel reminded me that's not protein. So it's of course the proteins that do the work and make things happen, and it's therefore important to see if there is a correlation between transcripts and protein. So if we look at the blood state, malaria parasites have robust daily rhythms in gene expression, and these rhythms are host independent. But there's about a delay of 11 or more hours between the peak of messenger RNA transcription and protein expression. And I think in the sporozoites, we don't really understand the transcription and translation dynamics so well, but it's probably fair to assume that this is also tightly regulated, and there is some evidence for that. So, for example, there are some messenger RNAs that are very abundant in the sporozoites, but the protein is not detected until later in the life cycle. And what they did now, they kind of cross-referenced a data set of sporozoite proteins expressed in the salivary glands. So, which I presume is maybe a database that's publicly available, or one that, maybe from an experimental setup they previously done. But they kind of cross-referenced that with the cycling mRNA, and they found that about 1/3 of the genes that cycle at mRNA level are also translated into proteins in the salivary gland stage. And that suggests that maybe some of the sporozoite messenger RNAs that exhibit these rhythms, or these circadian rhythms, are translated into proteins, and others maybe a little bit later as and when they were needed. Does that make sense? Oh, getting there.
Interesting. I mean, one of the comments, so I like the way this is all being pulled together, right? So we see the cyclic expression in the salivary glands. We see a cyclic expression in the sporozoites. We're sort of connecting them. They do go on ahead and show that, why are they producing it, early in the morning? Because 12 hours later is when the biting is going to occur. There's a 12-hour lag between transcript and protein. But I'm left with a couple of questions. My first one is, what if you just took mosquitoes that never had a blood meal, had no sporozoites in there? Does their transcriptome have a pattern?
So I suppose they would need to look at uninfected mosquitoes.
Yeah, that's what I'm curious. Like, what's the background? Like, is there this, because is this just, does the sporozoite's circadian rhythm of gene expression drive what's going on in the mosquito salivary glands? Or would the mosquito's salivary glands have this circadian expression over time without the influence of the sporozoites?
I would guess so, because they have to feed, right? And that's what.
Yeah,
Maybe,
I'm thinking a lot of this because what they're trying, I think the story they're trying to tell is this is all being driven by the sporozoites. I don't know. I don't think it's all being driven. I think maybe some of it's being driven. So I'd like to see a comparison of what does that circadian rhythm look like? What is the pharmacopoeia in the saliva of Anopheles stevensii mosquito that does not have sporozoites in the
And
cell?
actually, that's not mentioned at all. So that would be a really good experiment.
Where's reviewer number 2?
Okay, so now we're kind of moving into the mammal. So, I suppose I'm repeating myself a little bit. Let me just check the time, because I think, Vincent, you have a train to catch, so I better speed up a bit.
You got 15 minutes.
Yes. Okay. So, I think we said Anopheles mosquitoes are more likely to bite at night, and many of the salivary gland-specific genes are under circadian control. So that suggests that mosquitoes are prepared to have a successful blood meal at specific times of day. And they actually looked whether those blood meals were successful in figure 4, and in particular figure 4C. So what they show is that actually mosquitoes that feed at night, they seem to be, so it's figure 4C, D, and E in particular. Actually, that shows that they have they ingest more blood, they have higher hemoglobin levels, and that translates into also 30 minutes later, when they look at the midgut, they also have more blood ingested in the midgut. So that really shows that night feeding seems to be a more successful strategy in terms of what they can take up or what they can ingest. So I think that is quite nice. So I think that's nice. So that suggests that even if mosquitoes can feed anytime, they actually have a more substantial meal or a more successful meal during the evening. So because as their ability to successfully feed is also changing throughout the day. And so maybe actually that's what they suggest now. That's what you just said, Daniel, that these behavioral changes across the day may have created an evolutionary pressure for sporozoites to be also equipped with daily rhythm. So I think you said it the other way, Ryan, didn't you? You said they were sporozoite driven.
Yeah, I think they're good about, I think, being honest with what the data shows. So they in their discussion, actually right before the discussion, there's a line where they say, taken together, our results suggest that the circadian regulation in both the mammalian host and the Anopheles mosquito vector has driven Plasmodium to evolve rhythms that align with both hosts, ensuring successful transmission. Because we don't know, but now they do say in the discussion, our results lead us to propose that sporozoites have evolved to be rhythmic and align or exploit with the rhythms of vector and or host. And I guess I'm sort of left with, are they exploiting? Are they just kind of getting their timing right so they're in sync with everyone else and they're exploiting opportunity? Or is the transcriptionally active, rhythmic, transcriptionally activity of the sporozoites actually driving some of the expression of some of those, some of the pharmacopoeia in the mosquitoes?
Yeah, so I think the final kind of set of experiments brings it together quite nicely, really. So they went on to do a mouse infection experiment to see if the time of day affects parasite transmission efficiency independently of mosquito biting. So what they actually did, so this is all in vitro, so they didn't actually send out infected mosquitoes and let them feed on mice. So what they actually did is they dissected the salivary glands of infected mosquitoes at the beginning of the day and at the beginning of the night. So that's going to be making it very controlled. And they infected the mice with sporozoites by intradermal injection. So I suppose a pretend mosquito bite.
Yeah, it's interesting that they do that. I mean, I don't know why they didn't just let the mosquitoes bite the mice, right? Because they already did that early on. That's how the sporozoites got the mosquitoes to begin with. So there's obviously no issue. So why not just let them feed in the morning, middle of the day, at night, and then just compare? I'm not really sure why they did this.
Well, maybe it's just because that way they could do those mismatch experiments as well. I don't know. And also maybe it's just a bit more control in terms of amount of sporozoites. You'd have to kind of same number of sporozoites, I guess, for each of the infection experiments. And you don't really know how many sporozoites get injected when a mosquito feeds. I presume it's not always going to be 5. Or 7.
That's interesting. That's why I thought it would have made sense also to kind of line it up and see. But
so what they actually saw, then they obviously dissected those poor old mice, and they saw that there's a reduced parasite load in the livers of mice when the infection was initiated during the daytime using daytime sporozoites, compared to nighttime infections using nighttime sporozoites and mice. And then they kind of swapped the sporozoites around, and then they actually could see that this parasite load was abolished. So it seems to be very clearly a higher parasite load in the liver seems to be clearly associated with night feeding. So
I guess that's because the genes are all turned on at night, right? That you need for that, right?
Yeah.
Or they were all turned on 12 hours before, and now all the protein expression is there.
Yes, that's right.
Yeah.
And I think finally, they wanted to find out.
I feel like I'm hanging out with you too much, Vincent. I'm becoming a stickler.
That's fine.
Stickler for details.
Same thing.
It's all your fault.
It's a good effect.
It's not a fault. It's a good thing.
Yep.
And then really, finally, they wanted to kind of find out if the sporozoite cycling was also synchronized with the host. So they used liver transgenic liver cells that were expressing a bioluminescence reporter under the control of circadian clock. So I suppose they would kind of light up in 12-hour cycles. So they glow and stop glowing under circadian rhythm. So I think maybe with this, they maybe simulated nighttime biting by mosquito. I don't know. I'm not entirely sure, so maybe help me out. But in essence, is when the hepatocyte population was synchronous, so they were all cycling in the same kind of pattern, not just at random, randomly, and matched the time with sporozoites, there was a significant increase in infection at night. So I think the take-home message really is that essentially when they infected a nighttime mouse with nighttime sporozoites from a nighttime mosquito, the parasite load was higher than when they infected a daytime one with daytime sporozoites from a daytime mosquito. Am I right?
Yeah, they use like a luciferase reporter, so firefly luciferase, and they do light up, Yeah,
and that's actually seen in figure 4F, which is the final figure. And actually, you already read out the take-home message earlier, Daniel, but it might be worthwhile saying it again. So taken together, these results suggest that the circadian regulation in both the mammalian host, the Anti-Anopheles mosquito vector, has maybe driven Plasmodium to evolve rhythms that align with both hosts. So now they not only say, the sporozoites has kind of taken advantage and done a similar thing as the mosquito, but also aligned its transcriptome or its expression profile with the host. So that was the kind of final experiment. So I thought actually it was a really nice paper. It was very clearly written and the figures are really clear. I don't know, I found it actually quite pleasant to read, although it took me all day.
They say that maybe we could exploit these, we could disrupt this synchronization between vectors and parasites to try and disrupt transmission. I don't know how you would do that. What do you think, Daniel?
So one of the things they talked about was timing your insecticide spraying, sort of like targeting insecticide spraying during periods of increased mosquito activity. Yeah, I'm trying to figure out. The other is that maybe it's not so bad, and hopefully some of our listeners can maybe write in and comment about this, daytime biting thing. Is it that half of Stephen's eye have the ability to daytime bite? Is it really a preference? Is it evolving or changing over time? But maybe it's not so bad if they bite during the day. It may take many more bites in an urban center during the daytime. It may not be as big a deal because, you bring up, Vincent, like, is it that people are outdoors at night? And we worry about that in Uganda because with global climate chaos, which is a thing, it used to be that when everyone was home for the Christmas holidays, we had moved into the dry season. But now the wet season is continuing. And during that period, people are out late. Everyone is seeing all their friends. They're out of the house. They're not under the mosquito nets. And now it's a time of active malaria transmission. Yeah. It can be both.
So maybe you could modify the mosquito so that it's biting during the day and release those.
Yeah, genetically engineered mosquitoes that
we put Wolbachia into mosquitoes and release them.
We do, yeah. Vincent, you said that they suggest that this could maybe be used to kind of somehow mismatch the two rhythms with each other. Yeah. But I think for that, you really need to actually know what regulates those rivers because, that's what you'd really need to interrupt, wouldn't you? And before we actually have an understanding of what these regulators
we'll take
of the cycling are.
From America, from like the West, about 12-hour time off from Africa, and then we'll trade mosquitoes.
They'll get used to it.
We may have to do it like every year.
Yeah, but I think if you could chemically disrupt it, I don't know.
Oh, that might be interesting. Yeah. Some way that is not, because you don't want to, you want to, the ones that are disrupted that are no longer malaria transmitters, you want to allow them to outcompete the ones that are malaria transmitters. Yeah.
Yeah, but you don't want to disrupt human circadian rhythms, obviously. So whatever you do has to be specific for the mosquito.
Yeah.
Right.
Yeah.
There's one thing that kind of stuck out to me a bit that kind of I wasn't so sure about. So they kind of said that we learned earlier that the cycling also includes metabolic pathway components, and they kind of suggest that could be relevant for male or not infected mosquitoes. But actually, I did a quick and dirty online search, and that mentions that mosquito males primarily feed in the daylight hours. So I'm not quite sure if we can actually extrapolate the cycling to males. As I suppose it wouldn't really be relevant if they feed in the day anyway.
Yeah, we don't care about the males, right?
Yeah. So
now I'm thinking of if you spray for you put insecticides, you could put something else in the spray that would disrupt the mosquito circadian. You make a small molecule that would disrupt it. That could be useful.
So maybe that's something they're working on now to kind of identify those regulators, and then maybe next year we can talk about that.
Well, put a little Ambien in there so they all fall asleep at night.
That's right. What does Ambien
Wake up
do?
Hungry the next morning.
What does Ambien do?
Oh, it makes you sleep. Yeah, it's like
just
a sleep
so you can sleep anytime.
Yeah.
And I suppose
Valium, we could use Valium.
And I think another thing that would be really important to do is now actually look at you mentioned Anopheles gambiae, for example. I don't know if they're maybe not as easy to have in an insectary. I don't really know. We do have an Anopheles insectary in Glasgow, but I don't actually know which species that they're growing, but that would be something to find out. So I think that would be interesting to do that experiment with gambiae and maybe falciparum, because it should be possible.
Yeah, I'd be interested. I would like to know that.
Yeah.
And then I want to know without the sporozoites, like, do we see the same? I'm curious, like, is this driven, or is it just that the sporozoites are just getting in sync?
They're just kind of riding the wave.
Yeah. Are they riding the wave, or are they manipulating the wave?
Yeah. Anyway, I thought it was a really good paper, and I'm glad I picked it. Sometimes I choose a paper and then I somewhat regret because I don't understand it.
Great
I think that was a really nice paper,
presented,
well written. So I really enjoyed that. So that's it from me.
All right.
Yeah, I
All
think we
right,
probably don't have time for the hero. So,
we don't. No,
I'll
maybe
save that for next time.
Save it for next time. 2 weeks from now, right? Is it 2 weeks? Yeah.
Yes, I'll be in London, but I think I'll have probably
They have
got
internet in
Wi-Fi.
London. They have
They do,
internet.
yeah, they
I
do,
hope
yeah.
so.
But I might go to the ballet or something, or to the pub. No, I'll
Yeah.
be here. I'll be here.
All right, that's TWiP 260. You can find the show notes at microbe.tv/TWiP. You can send in questions if you'd like. We actually have a few emails we have to do sometime. TWiP@microbe.tv, and also your clinical guesses go to TWiP@microbe.tv. If you enjoy these programs, we would love to have your financial support. Microbe.tv/contribute. Daniel Griffin, Columbia University Irving Medical Center, parasiteswithoutborders.com. Thank you, Daniel.
Oh, thank you. And everyone, be safe.
I didn't ask you what's on your tie. It looks just black to me.
Well, so it is Friday.
Yeah,
and this is
sexually
an
transmitted.
HIV awareness bow tie with the red ribbon.
Red ribbon. Okay. Christina Nell is at Glasgow University. Thank you, Christina.
Well, thank you. That was a really fun TWiP, as usual. And Daniel, have a fabulous time in Scotland.
Oh, thank you.
Tell us all about it. And if you remember the name of the island you go to, let me know.
Okay. Yeah, I'll let you know.
I'd like to thank, I'm Vincent Racaniello. You can find me at microbe.tv. I don't remember, but twice is okay. I'd like to thank the American Society for Microbiology for their support of TWiP and Ronald Jenkins for the music. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP is parasitic.